Okay, can somebody confirm you can hear me? Boy, my voice is kind of hoarse.
I can hear you.
Yep, I can hear you.
So today is July 16th, and we're supposed to be starting chapter 2. We're a little bit behind schedule. I'm pretty sure we have to finish chapter 1. Let me see where we are. Yeah, we talked about the birth of modern chemotherapy. Okay, before we begin, are there any questions? So we were supposed to talk about chapter 2. We will get there. We've got about 5 more slides of chapter 1 to finish. Let me remind you, you have a practice quiz 00, which you must take by 11:59 p.m. on Wednesday. You can take it anytime between, well, let's say midnight or 11:59 p.m. Tuesday night, and then anytime Wednesday morning till 11:59 p.m. You should take the practice quiz to get yourself uh, familiar with my quizzes, as well as to test your Respondus Lockdown browser and make sure it's working for your system. If you wait until quiz 1 and then you find it's not working, by the time you contact the computer help desk and they give you instructions for changing your setting, the quiz may already be timed out. Any questions about any of that?
The practice quiz, is it regarding chapter 1?
Oh, I don't have that up there. I'm supposed to. Let me see if I've got that on an older syllabus. I think it's only chapter 1 and lessons in the lab module 00 and lab module 01. But let me check. And I need to update the syllabus on that. Spring 2024 will be the latest. Sorry, you asked me a question I just don't know the answer to, so I want to look it up. Yes, it's on chapter 1, obviously chapter 10, and lab 0 and lab 01, and then the syllabus. That is supposed to
Thank you.
be syllabus, but it's not. I took the schedule from the last summer, and apparently I hadn't had that on the syllabus last summer. There we go. Now, hopefully, I'll change your online syllabus, but all right, we'll keep that for later. Close that down. Let's go to chapter 1. So, the start of modern chemotherapy happened in 1910. You don't need to know the year, by Paul Ehrlich, discovering a drug which could cure syphilis. And he did that in 1910. Alexander Fleming discovered the first antibiotic in 1928. And sometime I'd like to give you the story of antibiotic and the first use of it, clinical use of it, but we'll wait until I have more time. Alexander Fleming actually discovered the antibiotic penicillin G, because he was a sloppy microbiologist And when he streaked out his plate, he got a contaminant of mold growing on his plate. And then instead of putting the plate in the incubator, he grew it on the counter. He just left it on the counter where he streaked it out. And then he took off for the weekend, came back on Monday and discovered the bacteria was growing, but he had a contaminant on it. And he noticed a ring around the contaminant. And he correctly concluded that something was diffusing out of the mold that was preventing or killing the growth of the bacteria. And he called this penicillium, which was later called penicillium, he called it penicillin with an N. It's coming from the mold Penicillium and with an M, and it's now called penicillin G, the first antibiotic. It was not the first antibiotic that became clinically available. That was actually a sulfa drug, sulfonamide. It was a man-made drug, and they were able to purify it because it was man-made, and then made it available clinically in 1935. Penicillin G, even though it was discovered in 1928, Alexander Fleming could not purify it. And it wasn't purified until the early 1940s, where they grew up the mold, harvested the penicillin, and then purified it, and then were able to inject it into patients. Any question about any of that? You might hear me using the term microflora to describe microbes. That is an older term that is no longer used. And I try not to use it, but it is the term that was used to describe microbes when I was taking microbiology, which was a long time ago. And bacteria were once classified as plants. If you remember, Carolus Linnaeus classified all living things into animals and plants, and bacteria were classified as plants. Hence the term microflora for our microbes. That has now been replaced with microbiota. When we're talking about the normal microbiota, that is the microbes that are normally present in and on a living organism. All animals and plants host a symbiotic microbial community that live with them, either in or on them. And when we're talking about the human normal microbiota, you should actually word it human or Homo sapien normal microbiota, because it's slightly different than, say, the microbiota of cats. For example, cats have Bartonella henselae as part of their normal microbiota. And this organism is not growing on humans. And in fact, if it does grow on humans, it causes a disease, and we call it cat scratch fever. Any question about any of that? So the normal microbiota are microbes found on and in the skin, the oral nasal cavity, the respiratory tract, the upper respiratory tract, the digestive, and the urethra. The normal microbiota is normally not found internally in internal organs and tissues. The normal microbiota can help prevent the growth of pathogens. They can compete for resources and for space. They can secrete toxins that actually inhibit the growth of pathogens. And then the normal microbiota can produce nutrients and growth factors. For example, humans get a significant portion of the folic acid and vitamin K from the normal microbiota growing in their digestive system. The normal microbiota is also very important in helping animals digest their food. Like E. coli helps us digest our food. Any question about any of that? So in the respiratory tract, we have normal microbiota, obviously in the mouth and the nasal cavity, in the throat, in the bronchi, but down deep in the lungs, we usually do not have normal microbiota growing there. And if microbes do grow deep in the lungs, we call that pneumonia. And that obviously is a disease. The normal microbiota do not normally cause disease. However, they can cause disease in certain circumstances, such as a patient who is immunocompromised and the normal microbiota, I don't know, overgrows or grows where it shouldn't, like in an AIDS patient, and then it can cause disease. Infectious disease results when a pathogen overcomes the host resistance, and this is caused, the disease is caused by a pathogenic microbe. An emerging infectious disease is a new or changing disease that is either increasing or has the potential to increase in incidence. Can anyone name a new and emerging infectious disease? Come on, you should know probably two.
COVID one?
COVID-19 is a new and emerging infectious disease because before 2019, it, as far as we know, never hit the human population. Obviously, it's now become a part of the, I don't know, the human population. So eventually it'll not be a new and emerging potential disease, but I don't know how long it'll take before they lose the nomenclature. Like HIV used to be called an emerging and infectious disease. And I don't know if that's still called that because that's been in the humans. Well, it's been in the humans for much longer, but we've known about it since the 1980s. So that's been like over 40 years. So I don't think it's called an emerging infectious disease anymore. So eventually COVID-19 will no longer be called an emerging infectious disease, but at the moment it's still new enough that it is. And then another one is bird flu. There is concern that bird flu could change, and then when a person gets it, it could spread person to person. If you don't know, bird flu is communicable from birds to people, but it doesn't spread from people to people. And so it's a emerging infectious disease, and they're keeping an eye on it. And at the moment, they're just worried that it could change. It may never change to spread from people to people. But like I said, it's an emerging and infectious disease, and they're keeping an eye on it. The reason why they're keeping an eye on it is, if you don't know, bird flu is fairly lethal to humans. I don't know what the lethality rate is, but it's something like at least 10%, maybe a little higher. All right, so that's all you need to know about emerging infectious diseases. I've got a few other slides here. I'm not going to cover it, and you will not be held responsible for this material on the quizzes, but it's talking about emerging infectious diseases in greater detail. Here, this one, they've got bovine spongiform encephalopathy, which actually is called Creutzfeldt-Jakob disease, at least in most humans, and mad cow disease in cattle. And that is an emerging infectious disease. And I think this really hit the news sometime in the 1980s when the British cattle all became, well, not all, but a large number of them became infected with bovine spongiform encephalopathy. So it sticks around the term emerging infectious disease for a while. And there's one, necrotizing fasciitis, the flesh-eating disease. That first made its headline, at least in the 1990s. And here they're still calling it an emerging infectious disease. The reason being is that its incidence has been increasing since 1995. I'm not sure why that's the case, but all right. Any question about emerging infectious disease? Like I said, you only need to know the definition. If not, let's close that down and begin chapter 2. Somebody has their microphone on. Could you silence your microphone? Let me try. Well, that didn't work. Stop the share. I'm just looking to see. All right, they've silenced their microphone. I think I'm sharing this, aren't I? Because I got stopped share there. Sorry, I didn't mean to show that, but fortunately the student who had their microphone on already shut it down. All right, chapter 2, chemical principles. The major goals and the rough outline is what I'm showing you now. Know the terms, always know the terms. And in fact, remember, you really need to know more than just the terms. You really should learn the objectives, which, let me remind you, you can get all of the terms under the objectives by scrolling down to the study guide and opening it up. And then the very first part of the study guide will be the objectives. And then later in the study guide, if I can get it, we have the chapter notes. And now that I've finished chapter 1, let me make the slideshow available to you. So if you want to add to your notes, you can. But please do take your notes because you'll learn best from your own notes. All right. And then some other topics we're going to cover is 2, with respect to the 4 major biological molecules, be able to describe the relationship and the characteristics of the monomers and the polymers, and then describe the composition, structure, and function of each of the 4 major biological molecules, and then give examples of each. What do I mean by the 4 major biological molecules? Can anyone tell me what I mean, or what are the 4 major biological molecules? Come on. You should be able to get 3 of the major biological molecules by looking at any cereal box or any ingredients on something that you eat.
Is it lipids, proteins, nucleic acids? I'm not sure what the fourth one is.
Carbohydrates. Very good. Yeah, you got you got carbohydrates, lipids, although we call it fats on the cereal box, and protein. You can find those in all list of ingredients with the food. But you stated nucleic acid, and that's one that is not on any food box, like a cereal box. So good job. All of these molecules, excuse me, get out of there. Come on. All of these four major biological molecules can be made up of monomers and polymers, although the lipids, some of them are made up of monomers and polymers, others are not. So others are just polymers. But the carbohydrates, the nucleic acids, and the proteins are all made up of monomers and polymers. In chapter 2, you should know the scale of things. You should know a human is about the largest thing we will discuss and study. A chicken egg is actually the largest cell you've ever seen. The yolk of the chicken egg is the largest cell I'm sure all of you have seen. And I've had one student who actually saw the yolk of a turkey egg, that would be even larger. And if anyone's ever seen the yolk of an ostrich egg, that would be the current largest cell on the planet Earth at the moment. And then about the smallest thing our human eyes can see are frog eggs. We can get a little smaller than that, but frog eggs. Below that, we have most eukaryotic cells, which are much smaller than a frog egg, which are much smaller than the yolk of a chicken egg. Below the size of a eukaryote cell, we have most prokaryotes, most bacteria. Below the size of most bacteria, we have the size of viruses, and they are small enough that you need to see them with something smaller than a light microscope, or not smaller, but able to see smaller than a light microscope, such as an electron microscope. Smaller than a virus are large molecules like proteins and lipids. Smaller than large molecules are small molecules like water. And then smaller than small molecules are atoms. Any question about any of this? You do not need to know the size range, but you should know the rank. A person's bigger than a chicken egg, bigger than a frog egg, etc. Okay? And let me state there is one error in this slide right there. It's got 1 micrometer there and 1 micrometer there. This right there should be 1 millimeter. Any question about any of that? All right, in chapter 2, we're going to talk about what are organic compounds. Organic compounds typically are more complex than an inorganic compound. They typically have carbon holding the molecule together. So carbon is a central part of an organic compound. And carbon is important because it can bind to 4 other atoms. And then organic compounds are held together mostly by chemical covalent bonds. They're not held together by ionic bonds or hydrogen bonds, although they can have these other bonds in the molecule. They mostly are held together by chemical covalent bonds. Any question about any of that? All right. An inorganic compound typically lacks carbon, but they don't always lack carbon. They are required by living organisms for the organism's growth, repair, maintenance, and reproduction. Some examples of inorganic compounds include oxygen gas or O2, nitrogen gas or N2, and then carbon dioxide or CO2, and then water, another important inorganic compound. You'll note that CO2 is an inorganic compound that obviously contains carbon. And I did say inorganic compounds typically lack carbon, but they don't always lack carbon. And then water is one of the most important and most abundant molecules in an organism. Even though it's an inorganic compound, it is the most abundant and one of the most important molecules in an organism. Any question about any of that? Water is a polar molecule. A polar molecule is one that has a charge differential on one end, so it's partially positive towards the hydrogen and partially negative towards the oxygen. And I don't think your textbook calls it a partial charge. I think it calls it a slight charge. What we're talking about is it is not a +1 on hydrogen and a -1 on oxygen. It's a partial plus charge on hydrogen and a partial negative charge on oxygen. The partial charge happens because the electrons, which form the covalent bond holding hydrogen to oxygen, and there'll be two of them, are pulled stronger towards oxygen and spend more of their time around oxygen than they do around hydrogen. That gives hydrogen a partial positive charge and oxygen a partial negative charge. Now remember, that is not a +1 charge or a -1 charge. It's a partial charge being something between 0 and 1. Any question about any of that? The polarity of water allows water to form hydrogen bonds with other water molecules, meaning this partial negative oxygen is attracted to this partial positive hydrogen. And that electrical attraction we call a hydrogen bond. And that actually is why water is a liquid. I don't know if you can see this water in here. I should have had a glass one, and then you can see the liquid in there. I don't know if you can see that very well, but this isn't water. It says sanitizer, but it's close enough to water. And it shows you it's a liquid holding together, and a liquid will take the shape of the container it's in. That's why we call it a liquid. The liquid is because these hydrogen bonds break and reform all the time, which is why water can take the shape of the container it's in. If the hydrogen bonds were to never break, the water would never take the shape of the container it's in. And we have water with hydrogen bonds never breaking. You simply freeze the water, and then we call it ice, and it doesn't take the shape of the container. And that's because the hydrogen bonds are locked in place. They don't break and reform. And when the bonds don't break and reform, meaning they're locked in place, we have a solid. We call that ice. If the bonds, hydrogen bonds, break and reform all the time, it is liquid water. And then you can heat liquid water up, and then you get steam, in which case the hydrogen bonds are broken. And then the water molecule doesn't stick to other water molecules. It goes flying out into the air. and that we call vapor. Any question about any of that? The important part is that water has two molecules. It has a chemical covalent bond, which is a polar covalent bond between the hydrogen and the oxygen within a water molecule. But water, at least liquid water, has hydrogen bonds between different water molecules, at least when water is a liquid. And I guess it does have hydrogen bonds when water is a solid also. The difference is the hydrogen bonds do not break and then reform. Any question about any of that? All right, there's a little video you can watch about the water molecule. I'm not going to show it because we are a little behind schedule. Water is a good solvent because all polar molecules can dissolve in water, and all ionic molecules can also dissolve in water. So that's why water is a good solvent. A polar molecule would be something like water, shown here, and obviously water can dissolve in water, but that's not the point. But glucose is a polar molecule, and it forms hydrogen bonds with water, and that is why glucose can dissolve in water. And you all know that because if you add sugar to tea or coffee, or you know that soda pop has sugar in it, it dissolves in the water. Well, ionic molecules like sodium chloride can also dissolve in water. Here, let me blow this up. Here we have a salt crystal, which is forming an ionic bond between a +1 sodium here, and then a -1 chloride ion here. And then this +1 is attracted to the -1, forming an ionic bond, forming the crystal structure shown here in salt, sodium chloride. Well, water can have the negative oxygen come up to the positive sodium ion, and then the negative water can pull out the +1 chloride ion, and then surround the +1 with a bunch of partial negative oxygen, where the water will have the oxygen oriented towards the sodium ion and the hydrogen's away from it. And then that will allow sodium to dissolve in water. And the same with the chloride ion. The -1 chloride ion will have water come up to it, and then the partial hydrogen will be pointing towards the negative chloride ion. The oxygen will be away from it, and a whole bunch of water can come up to the chloride ion, pull it out of the sodium chloride salt crystal, and then bring it into solution, and the hydrogen ions will be surrounding the chloride -1, and then that will make the chloride ion in solution. Any question about that? Anything about any of that? Let's watch a little video on that one. Yep. Having trouble getting that to play. Don't know what the problem is. It's supposed to be a link which I can click on, but it's not seeming to work. a water dissolves salt. So there's a water molecule, partial positive hydrogen, partial negative oxygen, a bunch of water stuck together, showing here. Water comes up to sodium chloride, a salt crystal. Oh, there's the positive, partial positive here and here, partial negative around the oxygen. And then a sodium is +1, a chloride a -1. That's a complete charge on the sodium chloride. Water comes around chloride, and then the positive hydrogen, slightly positive hydrogen, surrounds the chloride ion. Any question about any of that? And that's why salt will dissolve in water. Any questions about any of that? So water is a good solvent because it is a polar molecule. It can dissolve other polar molecules as well as ionic molecules. Polar molecules are hydrophilic. Literally, this means water, hydrophilic lover. What we mean by that is that the molecule is attracted to water and can dissolve in it. Non-polar molecules are repelled by water. We say they're hydrophobic. Literally, this means fear phobic of water, hydro, fear of water. The molecule that's hydrophobic does not dissolve in water. An example of that would be oil. As you all know, when you're making salad dressing, when you mix the vinegar, the water, and the oil all together, the oil molecules are not attracted to the water and they will disassociate from it. And the oil will not dissolve in the water. Any question about that? So hydrophobic doesn't literally mean fear. I mean, literally it means fear of water. But what we mean is it's not attracted to water. It doesn't dissolve in it. And the best example is oil. Although you can use any fat or lipid, almost any fat or lipid. There is one exception. We'll talk about that. Hydrogen bonding between water molecules makes water excellent at buffering the temperature. You guys should know this because you live in Western Washington or Western Oregon, and our temperature is buffered compared to Eastern Washington or Eastern Oregon. And the reason why our temperature is buffered is because the moisture from the Pacific Ocean buffers our temperature, making it less hot in the summer than in the eastern part of the state, and then making it less cold in the winter compared to the eastern part of the state. And it's simply because we have all that moisture from the Pacific Ocean buffering our temperature. The water buffering properties of water happen because water has two things that can absorb a lot of energy. So that when we heat water up, there are two things that can absorb that energy. First, it takes heat to break the hydrogen bonds and then make the water evaporate. So the different water molecules that have hydrogen bonds between them, when you heat them up, you break those hydrogen bonds, causing the water to evaporate. And not only that, but the hottest water molecules are the ones that evaporate. And so that takes the most amount of heat away from the water. And that's why water is actually good at cooling an individual, like when they're perspiring, because the hottest water molecules are the ones that evaporate. But the point is, it takes energy to break the hydrogen bonds, and that's one thing that water has to absorb that energy when we're heating water up. The second thing that can absorb the energy is that when the water molecule itself, or any any molecule, absorbs heat energy, it will absorb that energy and then cause the molecule to vibrate faster and more. And that faster vibration is actually the measure of the temperature, meaning something that is at a warmer temperature, a hotter temperature, will have its molecule vibrating, moving about faster than something that is at a cooler temperature. And that's actually what temperature is, the movement of the molecules in these, I don't know, the object or in the substance. So water has two things that can absorb that energy. Most other things, like other, I don't know, liquids, other solids, do not have two things to absorb the energy. Like when you heat oil, only the oil molecules can absorb the energy. And therefore, when you were to compare the temperature rise in 1 cup of oil compared to 1 cup of water, the oil will raise to a higher temperature than the water when you're giving the same amount of energy to the water and the oil. And that's simply because the oil can only absorb the energy with its molecule absorbing it and then vibrating faster, which itself is a measure of temperature. But water has two things to absorb that energy. Both the molecule itself can vibrate around faster, increasing the temperature, but the hydrogen bonds connecting the liquid water molecules together can be broken, causing that water molecule to evaporate. And that is why when you heat water the same amount with the same temperature, the temperature of water will raise less than the temperature of oil or the temperature of almost any other substance, because they only have one thing to absorb the energy. Any questions about any of that? All right. Water can split into, meaning a water molecule can split into other molecules. And on the surface of the Earth, normally H2O can split into H plus and OH minus. That is normally what happens when water splits apart on the surface of the Earth. You have to be at the temperature of the surface of the sun to get water, H2O, to split into the hydrogen ions and the oxygen ions. So if you're ever given this question, don't tell me that water, H2O, will split into hydrogen ions and oxygen on the surface of the Earth, because that doesn't happen on the surface of the Earth. You have to be on the surface of the sun for that to happen. And let me state that this is not including adding electricity to the water. If you add electricity to the water, you can get electrolysis and then split the H2O into hydrogen ions and oxygen ions or atoms. And that's not a normal circumstance because you're adding electricity to the water. When water splits apart into H plus and OH minus, remember the H plus is a proton, the OH minus with a -1 is a hydroxide ion. These create charged molecules in water that can participate in chemical reactions. It also gives rise to the pH of the water. Any question about any of that? Let's talk a little bit about the pH. You have increasing acidity when you have increasing H plus ions in solution, more H plus ions than OH minus ions. That's acid. And the pH is less than 7 when it is an acid. Some examples: tomato juice around pH 4, grape juice around pH 3, lemon juice around pH 2. There'd also be vinegar. And then stomach acid is about the strongest acid you're probably familiar with, around pH 1.5, although your car battery acid is around pH 1. The pH of acidity is between pH 0 and pH 7. pH 7 is neutral water. And neutral water is where, when H2O breaks apart into H plus and OH minus, the H plus ions equal the OH minus ions in solution. And that's a pH 7, where the H plus ions and the OH minus ions are equal in number. Increasing basicness or alkalinity is when the OH minus molecules in solution are more numerous than the H plus ions in solution. And we have a base when we have something like sodium bicarbonate added to water, or milk of magnesia added to water, or household ammonia added to water, and that's actually around pH 11.5. Also a bleach around pH 12.5. And then oven cleaner is the strongest base you guys might be familiar with, assuming you cleaned your oven, and that's around pH 13.5. Alkalinity ranges from pH 14 down to pH 7. Any question about any of that? All right, I'll talk about that when I get to there. Acid, bases, and salts. An acid is a substance that dissociates, giving up one or more H plus ions to solution. So if you have HCl and put it in water, you get hydrochloric acid, because the HCl will break into H plus ions and chloride ions, -1, in solution, and that will increase the H plus ions in solution. So there'll be more H plus ions than chloride ions, and the acidity will be someplace between lower than pH 7 to pH 0. Any question about acids? Base, your textbook says, is a substance that dissociates, giving up one or more OH minus ions. And then they give the example: sodium hydroxide can dissolve in water, dissociating into Na plus 1 and OH minus 1. That will release OH minus in solution, increasing the OH concentration over the H plus concentration and making the pH something over 7 but under 14. Any questions about any of that? All right, I am here to tell you that a base is also a substance that dissolves or incorporates H plus ions into it, pulling the H plus ion out of solution, like sodium bicarbonate, this is bicarbonate right here, can be put in water, in which case it'll absorb the H plus of water to make carbonic acid here, H2CO3. And that will blow the H plus out of the water solution, making the solution alkaline or basic, because there will be more OH minus in solution than H plus, because the H plus are pulled out of solution by the bicarbonate. Any question about any of that? So remember, a base is a substance that either gives off OH minus or a substance that absorbs H plus ions. And the best example is bicarbonate absorbs H plus to make carbonic acid. A salt is a substance that dissociates into cations. That's a molecule with a +1 charge or more positive charges. And anions, that's a substance that has a -1 charge or more than a -1, like -2 charge. But neither of these ions are H plus and OH minus. And that's because if it were to dissociate in H plus, we don't call it a salt, we call it an acid. And if it dissociates into OH minus, we don't call it a salt, we call it a base. But anything that dissociates into a positive ion and a negative ion is a salt. And sodium chloride dissociates in water into sodium +1 and chloride -1. So sodium chloride, which you already know, is a salt. However, magnesium chloride is also a salt because it will dissociate into magnesium 2+ and 2 chloride ions. Electrolytes are substances that dissociate in water, forming ions that allow the water to carry electricity. So seawater is an electrolyte because it has molecules in it that form ions in the water, such as sodium chloride, which can then carry electricity. And if you do not know, if you're swimming in the ocean or some salt lake, and you see a lightning storm move into your area, you should get out of the water because if the lightning were to hit the water, it could do serious damage to you if the lightning were to be anywhere around you. So not just nearby, but any place around you. The salt water will be an electrolyte carrying that electrical charge to you. Why you shouldn't swim in a salt water like an ocean. However, distilled water like fresh water lacks these electrolytes. And so in order to get the same damage as a bolt of lightning in salt water, that lightning bolt in fresh water has to be really close to you before the electricity can be carried to you. If the lightning bolt is far enough away, and I'm not sure where the cutoff point is, so I won't state it, but if it's far away and the lightning hits the fresh water, it won't damage you because a fresh water is not a good electrolyte. It does not carry electrical charges. However, salt water does. Any question about any of that? So an electrolyte is a substance that dissociates in water and allows the conductivity of electricity. Any questions about any of that? All right. Organisms need to maintain a fairly constant balance of their cellular acids and bases to remain healthy. In Biology 160, you probably learned the term homeostasis, and that refers to this concept that organisms must maintain a fairly constant balance of cellular acids and bases to remain healthy. If your blood or your body were to become too acidic, you would become sick and then eventually die. The same thing with too basic. If the body were to become too basic, you will first become sick and eventually you'll die. The pH is a measure of the amount of H plus ions in solution. Is that clear to everyone? The pH is a measure of the H plus ions in solution. If you have more H plus ions in solution, then you will be an acid and the pH number will be smaller. If on the other hand, you have less H plus ions in solution than OH minus ions, then you'll have a base and the pH meter will show that the temperature has dropped below the room temperature, which at the moment would be pretty high, something like 93. The exact pH is actually a measure of the negative log of the H plus ions in solution. So you can count up how much H plus ions are in solution, put that into the log of it, and then take a negative of it, and that's the pH. The pH scale ranges from 0 to 14. It is a log scale, meaning that if you change the pH by 1 unit, like pH 7 to 8, you're increasing the alkalinity by 10-fold. So if you increase the H plus ions by 10-fold, you would change the pH scale by 1 unit. Any question about any of that? Most organisms grow best around pH 7. So something like 6.5 to 7.5, or you could say pH 6 to 8. That is when most organisms grow best. We can have some organisms that grow at different temperatures, I don't know, different pHs, but the majority of them grow best around pH 7. Any question about any of that? All right, if there's no questions about pH and water, let's move on to talking a little bit more about organic compounds. Here we're looking at several organic compounds. And first you can see that they can be fairly simple, but there's very few organic compounds which are pretty simple. Most of them are fairly complex, like acetic acid or this phospholipid here. Organic compounds can be drawn in a stick figure where you just connect the molecules together or the atoms together this way. And they can also be drawn in the ring structure as shown here. Oops, let me block that up. I meant to blow it up, not to move it. In the ring structure, you don't put in the carbon and the hydrogen and the other elements unless where the point is, it is not a carbon. So wherever there's a point, there is a carbon. We don't have an example here. Wherever there is not a carbon, at the point, you need to put in what that atom is. So instead of having a 1 for that, you'd actually put in the atom right there. So if there's an oxygen connecting this carbon and that carbon, you put an O right there in between that line and that line. Any question about any of that? Oops. In the show here. All right, do not memorize these organic chemical compounds. We won't ever ask you for what an organic chemical compound is exactly, but you should note that organic compounds tend to be held together by carbon, which all of these are held together by carbon. And let's blow that one up again. Oh, that's too far. Actually, I don't have a picture of it, so let me scroll ahead and find one. There we go. This is the ring form, and wherever there's a point where there's a carbon, you don't need to label that with a carbon. And wherever we have a point, that's a carbon. And see, this one here has an oxygen holding this carbon to that carbon. You put in the oxygen there. Any question about any of that? Trying to return to where we were. For organic compounds, the simplest organic compounds contain carbon and hydrogen. And those we call a hydrocarbon. And you can see right here a bunch of hydrocarbons, methane, ethane, propane. Hydrocarbons, of course, burn. And all they are hydrogen and carbon. However, organic compounds can contain other atoms besides carbon and hydrogen. They may also contain oxygen, amino acids, carbohydrates, nucleotides, and lipids. Can also contain various hydrocarbons. Shrink that a little. All right. Carbon can covalently bond with up to 4 other atoms, which is one reason why organic compounds typically are held together by carbon. And here we're seeing carbon here binding to 4 other atoms and molecules. This makes carbon the skeleton backbone for the organic molecule, where the chain is held together, like this one here, by the carbons in the organic molecule. You can also have functional groups bound to an organic compound, and that will be more than just a hydrogen. A functional group can be like an OH group. The functional group influences the chemical properties of this molecule, giving it different properties than CH4 or methane gas. And so the functional groups are important in the biological molecules because each of the monomers of a biological molecule as functional groups. The functional groups bind to a carbon backbone and influence the chemical behavior of the organic compound. For example, methanol has different properties than methane, and the only difference is methanol has the OH group attached to it. One property that methanol has that methane does not is that methanol can easily dissolve in water and methane cannot. Any question about any of that? And that's a property that comes out of the functional group. So the functional groups are responsible for most of the chemical and physical properties of a particular organic molecule. You can also have hydroxyl group, OH, in alcohols, and it is hydrophilic, so that the hydroxyl group tends to attract water molecules to it and then help that molecule dissolve in the it says alcohol, but it should be water. So in this table and the next one, I'm trying to show you the functional groups. The functional group here is NH2 and there OH. It'll always be bound to a carbon, and that's what we mean by R. R represents the remainder of the organic molecule here. And so R can be essentially anything. But OH and NH2 are functional groups, and they give the molecule different properties than if the molecule were missing these functional groups. You need to know the functional groups that are surrounded in red here, because you may be quizzed on these. The other functional groups we're not going to talk about, like a ketone or a methyl group. Come on. Two other functional groups you should know are carboxyl, that is the C bound to a double O, and then the COH group, that's the carboxyl group. Another group you should know is the phosphorus group, that is P and then O4. Usually this will have a -1 charge, but in this case it's got a -2 charge. Why do you think you need to know phosphate group, the carboxyl group, the amino group, and the OH group for your quizzes? Anybody have a guess? Why do you need to know those functional group and not the others?
Because they're part of the 4 biological groups or the properties that we talked about at the beginning.
Yes, very good. You can find these functional groups in all of the biological molecules, or I should say, you can find them in the biological molecules. And so you need to know these functional groups. OH, which is a functional group of an alcohol, we can find it in lipids and carbohydrates, as well as other molecules, like it is in glucose, it's in methanol. For the amino group, we tend to find that in proteins and then the monomers of that protein. And that would be something like glucose has, well, glucose has an OH group. An amino group can be found in proteins and in amino acids. And an amino group is just NH2 bound to the rest of the organic molecule. Come on. The carboxyl group is found in organic acids, which I'm sure you don't know too much about, but it's also found in lipids and proteins. And so you must know this molecule for that reason. And then phosphate, and another functional group in an organic chemical compound. Phosphate is found in ATP, DNA, and RNA, as well as the nucleotide, which I guess ATP is a nucleotide of DNA or RNA. All right, any question about the functional groups? Frequently, more than one functional group can be found in a single molecule. Like, for example, an amino acid is shown here. We have at least 2 functional groups for every amino acid. You have NH2 being an amino group on that amino acid, and then COOH or COH is the carboxyl group. And both of these are found on a amino acid or found in proteins. Any question about any of that? All right, if not, let's talk about the organic chemical compounds, how they make the 4 biological molecules, and how they tend to be made up of small organic molecules that were combined into a large macromolecule. Like starch is a macromolecule and a polymer of many glucose molecules. If you want starch, all you need to do is hook the glucose molecules together, one after the other, and then you will get we will get a polymer if they're hooked together. The smaller organic molecules can be combined to make the macromolecule. For example, glucose is a small organic chemical compound, and you can combine lots of glucose together to make a large macromolecule that we call starch. And all starch is a repeating unit, glucose, repeated one after the other. So macromolecules are polymers consisting of many small repeating molecules that we call monomers. Come on. So many of the polymers are made up of smaller units that we call monomers. Example would be all the complex carbohydrates, all of the amino acids, all of the nucleic acids like DNA and RNA is made up of a smaller monomer, and that monomer would be a nucleotide. Any question about any of that? All right. When we have an organic chemical compound, we can combine smaller molecules like these 2 monomers hooked together to make a polymer. And that will have the R right here break with the OH, and then the H is broken off this monomer here, and then that R will directly connect to that R, being RR, the polymer, and then the H will get together with the OH to make water. And that is a dehydration synthesis, where 2 smaller molecules come together to make a larger molecule, and then water is made. Dehydration synthesis. So synthesis of hydration, which would be water. You can go in the reverse reaction, take the polymer, split it with water. The water will break apart, part of it going to this R group, and the other part of it going to this R group. And then you split the polymer into the various monomers. Any question about any of that? Oh, I forgot to mention that last reaction, splitting the larger molecule, the polymer, into monomers, is called hydrolysis. And you should be familiar with it if you've seen Back to the Future. I think it's Back to the Future 2, where Marty McFly says to his mother, "Ma, you sure know how to hydrate a pizza." What she did was she got this hockey puck, put it into her hydrolysis machine, hydrated the pizza, and out came this beautiful looking pizza. Hydrolysis, converting the polymers by splitting water, adding water to make the monomers. Any question about any of that? All right, all of the carbohydrates, the proteins, and the nucleic acids are made by dehydration synthesis, combining the monomers to make the polymer. And then some of the lipids are made that way, where you combine the monomers to make the polymer. But not all the lipids have monomers. Any question about any of that? All right, so this is looking at it in a different format where we have a monomer coming into a polymer, and we make a bigger polymer when this monomer is added. This would be something like glucose and glycogen in your liver, and then you're eating glucose and more glucose is added. So you're adding glucose to the polymer, and then water comes off. Hydrolysis would be the breaking off of this glucose molecule, the monomer, by adding water, and then the water splits apart with the OH group adding to the monomer, and then monomer would be released into the blood, meaning glucose would be released into the blood. And then in the liver, we'd have a polymer. It's shorter than it used to be, but it's still a polymer. Any question about any of that? All right, the former, whenever you're making something larger, we call it dehydration synthesis or a condensation reaction. Whenever you have a large molecule and you're making something smaller, we do that by hydrolysis. All right, carbohydrates include sugars, starch, and other complex carbohydrates. Carbohydrates are an important energy source. Our cells use carbohydrates for their energy needs, but our cells also use carbohydrates for their structure and in making nucleic acids. Carbohydrates consist of carbon, hydrogen, and oxygen. However, it's more specific than just any carbon, hydrogen, and oxygen. And a carbohydrate has the general formula. For each carbon, there's 2 hydrogen and 1 oxygen. And then you just repeat that for the molecule. For example, glucose will repeat this 6 times, meaning that N would be 6, so that glucose has 6 carbons, 12 hydrogens, and 6 oxygens. Any question about any of that? All right, simple sugars are the monomer of a carbohydrate. And you can simply link those simple sugars together to make a string of single sugars to make the polymer. You can classify carbohydrates into 3 major groups based on their size. The smallest is a monosaccharide, and it only has 1 unit. So, yeah, it's got carbon in it, but there's only 1 molecule, and that would be something like glucose. A disaccharide, on the other hand, is two monosaccharides linked together. Lactose is a disaccharide, and you have beta-galactose and fructose. You have fructose and beta-galactose hooked together. Is that right? No, it's not fructose, it's glucose. Glucose and beta-galactose make lactose. So lactose is the disaccharide. And then you have polysaccharides. These are composed of many monosaccharides. The easiest polysaccharide is starch, which is a polymer. All starch is glucose hooked together one after the other for generally a long non-branching string of sugars. Any question about any of that? All right. So carbohydrates, we mentioned the monosaccharides. They are simple sugars. All of them have between 3 and 7 carbon atoms. However, most monosaccharides have either 5 or 6 carbon atoms, like glucose and fructose, beta-galactose are all monosaccharides, and they're all 6 carbons. Ribose and deoxyribose are the only simple sugars or monosaccharide that I know of that have 5 carbon atoms. The monosaccharides or simple sugars are all sweet, and they're all dissolved in water. So we say they're all water soluble. And remember, we have a term for that. We call it hydrophilic. The disaccharides are the joining of 2 monosaccharides, like this glucose here and that fructose, can be linked together by dehydration synthesis into sucrose. And we get the OH pulled out of the glucose, the H pulled out of the fructose to make water. And then this carbon here will directly link to this oxygen here, which is shown right there. So this is the oxygen of here, and then it now binds directly to this carbon of this glucose chain. So disaccharides are the joining of 2 monosaccharides and dehydration synthesis, and you can break down a disaccharide by hydrolysis. Like when we eat table sugar, shown here, sucrose, we can break it down by hydrolysis, adding water, which will then split, adding an hydrogen group to the fructose and an OH group to the glucose. Any question about any of that? Oops, went to the wrong one, I think. Went to too many wrong ones. Sorry, I meant only to go down one, and obviously went down many. I'm not sure why that happened, because I didn't use this scroller over here. I used that scroller over here. Oh, I see. It does work that way. All right. Polysaccharides consist of many monosaccharides. For example, starch, all it is glucose hooked one after the other, mostly in a single unbranching chain. It can occasionally have a branch, but mostly it's a single unbranching chain. Glycogen is similar to starch. All it is glucose hooked one after the other, but glycogen has many different branches. And so it's more branching than starch. Here we're looking at some starchy food. I think everybody knows bread, cereals, pasta, rice, and potatoes are starchy foods. What you probably don't know is that beans and chestnuts are about 1/3 carbohydrates. So if you did want carbohydrates, actually beans and chestnuts are a possibility. Now it is true that with beans, about 1/3 are carbohydrates, 1/3 of the weight of the molecule are protein, and about 1/3 of the weight of the molecule are lipids or oils. So beans are a starchy food, but they also have a lot of protein and a lot of lipids. Most people think of beans as a protein, but in reality, beans are both protein, carbohydrate, and lipid. And I assume chestnuts are the same, although I don't know the exact makeup of chestnuts. Yep, why did we go backwards? All right, this slide is showing you the different polysaccharides. We've already talked about starch. What did I do with the... There it is. Mostly glucose hooked one after the other without branches, although there can be an occasional branch, like right there. A glycogen is very similar. The difference is that the glucose is one after the other, but the chain of glycogen can branch much more than glucose. Cellulose is similar in that it's made up of a whole bunch of carbon hooked together, one after the other. And here we're seeing this purple line. The purple line stands for a glucose hooked together in a covalent bond to another glucose hooked together, etc. And then there will be repeating chains of that. And these chains are going to form hydrogen bonds between the glucose. So this glucose here, hydrogen bond to that glucose, as well as to that glucose there. Any question about any of that? So cellulose is similar to starch. The difference is that cellulose has a whole bunch of covalent bonds in the molecule. And that's why people cannot digest cellulose, as well as most organisms cannot digest cellulose. The few that do, like the termite, doesn't actually digest the cellulose. What it does is it has a stomach, and inside the stomach, it has a fungi, and the fungi, which is a mold, will digest the carbohydrate in the sugar plant. And then it makes that sugar available to the I lost the name of it. What is that insect called? The termite. We'll talk about peptidoglycan later in the term. For now, all you need to know is that it is hooked together with sugars. And so it is a partial carbohydrate. However, it does have short peptides in it, so we will talk about it later. It's more than just carbohydrate. It does have short peptides, and we'll just talk about peptidoglycan in a later lesson. Any questions about any of that? If not, let's move on to lipids. Lipids consist of carbon, hydrogen, and oxygen. But unlike a carbohydrate, lipids can be in different ratios of the carbon and the hydrogen and the oxygen. Furthermore, lipids do contain oxygen, but they contain very little oxygen. Let me show you a molecule. Come on. Here's a lipid here, right there. I blow this up. And you can see in this molecule that they're mostly carbons and hydrogen. Even here is mostly carbon and hydrogen. There is an oxygen right there and an oxygen right there, and that's it. So not very many oxygen in the lipids. Any question about any of that? Lipids are mostly nonpolar and insoluble in water. So we say lipids are mostly hydrophobic. There is one exception, and we will mention it. For now, I'll just mention that the one exception is not all nonpolar and not insoluble in water. And we'll talk about it later. It's a phospholipid, and one end of the molecule is hydrophobic, and the other end of the molecule is hydrophilic. But most lipids are nonpolar and insoluble in water, so we call them hydrophobic. You can use lipids for the structure of cells and an organism, as well as energy storage. Lipids are the primary component of cell membranes. We can classify lipids into three different categories: a simple lipid, complex lipid, and a steroid. A fatty acid is a simple lipid. It's a major component of lipids, and it consists of a chain of carbons, actually a chain of hydrocarbons. Let me blow this up. So here we're seeing a fatty acid. It's mostly carbons and hydrogen. The exception is right here and right there. This OH will come off when this fatty acid will bind to a mother molecule. But fatty acids do contain oxygen, but not very many, and they're all right here. And so we say that they contain the fatty acid 1-COOH group. Most other locations on the carbon, all you have are hydrogens. If the hydrogens totally cover the lipid or the fatty acid, then we call it a saturated fatty acid. So a saturated fatty acid is one that has all of the hydrogens and one that has only a single carbon covalent bond between two carbons. So this right here is a saturated fatty acid. An unsaturated fatty acid, on the other hand, has at least one double carbon bond holding the carbon groups together, or the carbon atoms together. And when you have a carbon with a double carbon bond, it will have fewer hydrogens attached to it. Let me show you that now. Blow it up a little. So right here, we have a double carbon bond. And you can see, because there's a double carbon bond, there is not an H here and an H there. Wherever there's a double carbon bond, you can sort of see it, there will be a kink in this molecule where the molecule will bend. all you need to have an unsaturated fat is to have one double carbon bond in the molecule. And this right here is an unsaturated fat or unsaturated lipid. If on the other hand, that were to have a hydrogen here and a hydrogen there and only one single carbon bond there, which is kind of what's shown up here, then that would be a saturated fatty acid. Or a saturated fat, sorry. This is a fat. Unsaturated molecules are better for our body because they are better at dissolving in the blood. They also have a kink in it so that the molecule cannot easily come together and form like a clog in the blood vessel. But it could be just a clog, doesn't have to clog the blood vessel. Could be a clog any place in the body. All right, so that's an unsaturated fatty acid and a saturated fatty acid. If ever there's only one single carbon bond, it's saturated, and that applies to the whole molecule. If there's one double carbon bond, and this one only has one, then this is an unsaturated molecule. All right, a simple lipid is a fatty acid or a fat or a triglyceride. You can combine the fatty acids shown here. Let me blow that up. Combine this fatty acid with the glycerol, in which case the H comes off of this OH group and the OH comes off of this fatty acid group. And you do this 3 times, meaning there will be another fatty acid binding right here and a fatty acid binding right there. You then get a triglyceride, which we call a fat. And then there will be a water for here and a water for here and a water for there. So a triglyceride is 3 fatty acids combined with one glycerol. A triglyceride or a fat is the body's richest energy source. We store this down as adipose in animals. Any question about any of that? You can just look at your belly. I'm sure everyone has some fat cells there, and you can look at a triglyceride. Triglycerides are the most common fat in the body, although another common fat in the cell is phospholipid, the phospholipid, and that's in your cell membrane. Because a fat or triglyceride will be the body's richest energy source, if ever you needed a bunch of energy, because you're going to go walking in some place like Point Barrow, Alaska, when your car breaks down, you got to walk 20 miles into town. What you going to do is carry as much triglycerides as you can, because when you eat it, you'll get the most energy from that weight of material. If you were to carry the same amount of weight in carbohydrate or protein, you will get energy out of it, but it will not be the most energy you could for that weight. Is that clear to everyone? I'm just trying to show that lipids contain the most energy. And so if you needed to carry something to get the most energy you can get out of it, that should be a lipid. That may not be the healthiest thing for you. Like if you were to carry 20 pounds of lard on your trek and you eat it to stay warm, well, you need it to stay warm, but it's not going to be necessarily healthy for you. It'd be far healthier to eat 20 pounds of carbohydrates or 20 pounds of protein, but you will not get as much energy out of it. All right, any question about any of that? All right, let's talk about the last, well, not the last lipid. Another lipid is the phospholipid. This is the main lipid in the cell membrane. The phospholipid is unusual because, as shown here, it has a fatty acid tail, actually two fatty acid tails, and then a glycerol right here. But in the third position of that glycerol, you don't have a fatty acid, which you would have in a triglyceride. You have a phosphate group. You'll note that this phosphate group is charged, and that makes this phosphate portion of the phospholipid here hydrophilic. This phosphate group likes to dissolve in water. The fatty acid portion of the molecule likes to dissolve in lipids, meaning it's hydrophobic, and this is hydrophilic. And actually, the glycerol part is also hydrophilic. You'll note that there is an R attached to that phosphate group. That R just stands for some organic molecule. It can be different things depending on the species and what somebody's eating, but it's just, we're going to put it in R and not talk about it. If you get a bunch of phospholipids and put them in water, they'll start lining up like this. And then they'll start lining up like that inside the water. And you'll note that when they do this, they will have the phosphate heads, which are hydrophilic, facing the water on the outside. And then on the inside, they have the heads, phosphate heads, facing the water on the inside. And then the hydrophobic tails are inside this liposome-like structure of the phospholipid. Any question about any of that? This liposome should be familiar to you because what does it look like? What looks like this? Come on, somebody has to know. It looks like a cell. On each of our cells, like your blood cell, you have a lipid membrane, and which we call the plasma or cell membrane. On the outside of that cell membrane will be the phosphate heads of the phospholipid, and that's because in the blood it's mostly water, and you want the phosphate portion of the phospholipid facing the water because it's likes to be around water. The lipid bilayer will be on the inside of the cell membrane because it does not like being around lipids. And then here on the inside of the cell membrane, once again, we have the phosphate heads of the phospholipid because inside the cell is mostly cytoplasm, which is mostly water. Any question about any of that? This is just showing you, you don't really need to know this, but trying to show you the difference between a cell membrane, which is totally saturated, which could be largely our membranes, and then one which has unsaturated tails. And something like this could be something like a moose around its cell membrane near its feet will be unsaturated because the feet of the moose will be colder than near the heart of the moose, which will be mostly this in its cell membrane. And it'll be this to keep the cell membrane more liquid when it is colder temperature around the feet. And you don't need to know much about that. But let me state that our cell membrane has cholesterol in it, shown here. And that's because our cell membranes don't have a cell wall. And all animals are actually this way, where they put cholesterol in the cell membrane. The cholesterol gives strength to the cell membrane. So the cell membrane is stronger than this cell membrane just because it has cholesterol in it. Any question about any of that? And that's our cell membrane as well as all animals. All animals put cholesterol in their cell membrane to give the cell membrane strength. All right. If there's no questions, I think I'm going to stop here for the lecture. And then I'll see you at, I believe, 7 o'clock for the lab. Let me check on that time. Make sure I get that right. Yeah, I'll see you in the lab at 7 o'clock. All right, if there are no questions, I'm going to log out and say goodbye. Bye.